Joining us for the October seminar on physical genomics. My name is Benjamin Keene. I'm the Senior Program Administrator here at Center for Physical Genomics and Engineering at Northwestern University. We're really excited and honored to have our invited speaker today, Dr. William Greenleaf, who is professor in the genetics department at Stanford University. Dr. Greenleaf's highly interdisciplinary research links molecular biology, computer science, bioengineering, and genomics to understand how the physical state of the human genome controls gene regulation and the biological state. Efforts in his lab are split between building new tools to leverage the power of high-throughput sequencing and cutting-edge microscopies, and bringing these new technologies to bear against basic biological questions of genome and epigenomic regulation. Since beginning his lab, he's been named a Ellison Foundation Young Scholar, a Baxter Foundation Scholar, Chan-Zuckerberg Investigator, an Arc Institute Innovation Investigator, and he's received the prestigious NIH Director's Pioneer Award. Dr. Greenleaf, thank you so much for taking the time to share your work with us today. A quick note for our audience, we'll have Q&A session at the end of Dr. Greenleaf's talk. You can also just type questions into the chat in the Zoom at any time, and we can address them at the end of the talk. If you want to learn more about the research and training programs here at CPGE at Northwestern, you can visit our website at physicalgenomics.northwestern.edu, or look at our video archive on YouTube, or you can follow us on Twitter, X, or Instagram. So now I can turn the screen share over to Dr. Greenleaf, and we can get started. Thank you once again.
Okay, great. Can you guys hear me and can you see what's going on?
Looks and sounds perfect.
All right, well, thanks so much for that very kind introduction, and thanks for inviting me to talk to you today. So I have to apologize. I'm definitely coming down with something, so I'm going to be coughing a little bit through this presentation, but hopefully my voice won't give out. So I apologize already. I'm not totally. I kind of went back to doing these in person, so I'm not used to the Zoom. Like it's like giving a talk into a well or something. So it's a bunch of black screens, but hopefully everybody's still there. Okay, so my lab is really fascinated by how let's see, this 2-meter long object, the human genome, which is folded and compacted and sequestered into a 5-micron nucleus. How this object encodes functional biomolecules that somehow interact with the genome itself through physical interactions of binding, and bring about the incredible phenotypic diversity we observe in the human body, from stem cells to bone cells to endothelial cells. So how is this possible? And so my lab is really interested in developing methodological platforms to attack this sort of grand challenge problem of how the genome evinces biological state. So maybe we split this into sort of a few different areas, trying to understand the molecular basis of genome functions. So how do cells know who they are? What regulatory elements and transfactors drive cellular states? How do these regulatory elements work at the molecular level through binding of transcription factors and then interactions with promoters and things like that? And then can we engineer accurate cell states with sort of programmed overexpression of master regulators? My lab has another arm where we like to try to repurpose high-throughput sequencing instruments to measure molecular sequence structure-function relationships across very large swaths of sequence space. Unfortunately, I won't have time to talk about that today. I'll just focus on the first two of these sort of interests of the lab. So every cell in your body has the same genome, but obviously brain cells are quite different from blood cells. And how could that be? Well, different cells sort of turn on these different landscapes of regulatory elements. People refer to these as enhancers, which then drive different gene expression programs. This is a very hand-wavy view of regulation. All the quantitative aspects of this are sort of still to be understood. But we do know that when regulatory elements are not regulating gene expression in a cell type, they're often tightly bound up by chromatin and unable to be bound by sequence-specific factors like transcription factors. Whereas if they are regulatory, then they have they're often in these elements of more accessible DNA. There's more loosely packaged chromatin, and interactions with transcription factors are allowed. So this lens of chromatin accessibility is a really valuable way to try and understand which elements are regulating in cell types and which ones are not. So my lab, in collaboration with Howard Chang, driven by Jason Witten-Roaster, who's now at Harvard, developed ATAC-seq. We call it ATAC-seq, not ATAC-seq. ATAC-seq, assay of transposase accessible chromatin, where you isolate chromatin in sort of intact nuclei. You expose these nuclei to TN5 transposase, which goes in and inserts sequencing adapters into regions of the genome that are accessible to the machinery of transcription, to be elements that are capable of being bound by transcription factors, for example. Then you immediately isolate these sequenceable fragments, amplify them, and then sequence them into the genome, and you get pileups in regions of accessibility. It's a very straightforward assay. The library prep and the assay are sort of the same step. It's so easy, I like to say, I used to say that it's so easy that a grad student can do it, but now I say it's so easy, even a PI can do it. Jokes on Zoom don't seem to work. Okay, so we can also look underneath the accessible elements and look for example, transcription factor motifs and infer the presence of those motifs that might be driving accessibility. So we can do this actually at the level of individual cells as well. And you get data that actually mirrors, in a lot of ways, the single-cell RNA-seq data. So I'm showing here on the left the classical UMAP, which is sort of a way to show 20,000 dimensional data in 2 dimensions. For the RNA-seq structure of, in this case, bone marrow cells and peripheral blood cells over here. And you can see here's the hematopoietic stem cells differentiating into erythroid or monocytes and into B cells. And if we look at the open chromatin landscape using the same sort of style of projection, we see basically a very similar picture. But so on the left, you can imagine this as the what of the molecular phenotype. And on the right is at least an aspect of the why, which are the regulatory elements that are driving these gene expression states. And so I claim that this is a very synergistic view of molecular features of these cells. It's a lot like the synergy between gin and tonic. These things are better together, I claim. Okay, so what can we do? We can, for example, look at a differentiation trajectory starting from hematopoietic stem cells here and going all the way to B cells, and ask what regulatory elements are opening across this sort of pseudo-time trajectory. And you can see incredible dynamic features of the accessible elements in this genome, some hematopoietic stem cell specific features, these early progenitor features, and then elements that are becoming accessible only in these plasma B cells. And you can look, for example, here at these elements, which are all about 500 base pairs in width, opening sort of in dynamic time across this differentiation trajectory around this RAG2 gene. We can also ask more global questions about motif accessibility within these regulatory elements and ask, for example, is the GATA2 motif more accessible early or later in this differentiation trajectory, and see, for example, GATA2 motifs become accessible here in hematopoietic stem cells and early lineages, whereas other motifs like FOXO1 turn on later in this differentiation trajectory, giving us some insight into which factors are actually driving this sort of phenotypic transformation of differentiation. So here's an example of deploying these single-cell methods in the context of human fetal brain development. So my lab, in collaboration with Sergiu Pasca, collected these human fetal brain at post-conception week 16 to 24, and then did single-cell ATAC-seq and RNA-seq on them independently. And here's our sort of peanut butter and jelly plot of these features. You can see, just to orient you really quickly, because we'll go through this data pretty fast, here are sort of the early progenitors, neuronal progenitors down here, and they differentiate this direction into more fully differentiated cell type neurons. That's in the RNA-seq space, and you see a very similar pattern here where these early progenitors differentiating here into more mature neurons. Okay, now what can we do? we can make a pseudo-time trajectory of this differentiation path from early to more mature neurons, and then ask, on the left here are regulatory elements that are turning on early versus late in this differentiation. And on the right are genes that are turning on locally that are close to those regulatory elements and putatively potentially driven by the accessibility changes of these elements that turn on, that are on early in these progenitors and then turn on at different points along this trajectory. We can do the same thing. We can ask about transcription factor expression over here, and then the motif activity scores, that is to say, how much chromatin accessibility there is associated with the different transcription factor motifs. You can see strong correlation between the expression and the chromatin activity of these different transcription factors, thereby nominating which factors are actually turning on and driving these cell state changes at different times. Now, what we can do, actually, in collaboration with Anshul Kundaji at Stanford, we can train deep neural network models that are tasked with predicting the chromatin accessibility that we have observed in these different cell types based on the genome sequence. So it just takes in genome sequence, and it's tasked with trying to predict chromatin accessibility. Amazingly enough, even on unseen regions of the genome, these models can actually learn the sequence features that bring about chromatin accessibility. As you can see here, here's the predicted chromatin accessibility track based just on the sequence in this cell type. And here is the observed chromatin accessibility. So it's really capturing a lot of these features. But even more interesting, these are actually interpretable deep learning models. So we can ask the model, which bases do you think are really important for establishing chromatin accessibility at these different elements? And then we can, for example, call the different motifs here. FOX1, a one cut, and another FOX motif seem to be the ones that are driving chromatin accessibility in this peak, for example. So we have a sort of base pair resolved view of how accessibility is being driven. And even more interestingly, we can use these models as sort of a sort of in silico oracle, where, for example, if the model thinks that CTCF is really important, we can install a de novo a de novo mutation to the genome and ask if that base were to be mutated, how much would it change chromatin accessibility? And in this case, if you change this C to a T, you totally ablate this motif, and the model predicts very strong changes in chromatin accessibility. So this is a way of nominating causal de novo mutations, for example, in non-coding regions, just by installing them into the genome and asking how much does this specific non-coding mutation, how much of an effect on chromatin accessibility is it going to have? So, for example, we can do this in a cohort of autism spectrum disorder patients versus controls. Now, if you just look at the total number of de novo mutations in these individuals' genomes based on trio sequencing, there's about 42,000 in both the controls and the patients with autism. So we're really looking for sort of needles in haystacks here. If you overlap these mutations with just chromatin accessibility, you don't really get any enrichment. But if you instead use these BPNet models to identify which mutations are highly impactful on the chromatin architecture, suddenly you see almost a twofold enrichment for cases over controls. So we think we're identifying causal mutations here that are in autism patients that are impacting important regulatory elements, blowing up their ability to bind the transcription factors and thereby create chromatin accessibility, and also impacting gene expression causally that actually cause this this disease. And if you do the same kind of thing, if you overlap these mutations in fetal open chromatin landscapes, or in models trained on, in fetal heart open chromatin landscapes, or in models trained on fetal heart, you don't get any enrichment. So this is a sort of model specific to the brain kind of effect that we're observing. Okay, so that's an example in normal human fetal development. Can we actually use these sorts of approaches to try and understand other diseases like cancer? So in this case, we partnered with the Cancer Genome Atlas to generate open chromatin data across 73 different tumors and 10 different cancer types. And if we do this sort of projection of the open chromatin landscapes, we see here these immune and stromal cells all cluster together from a lot of the different tumors. But oftentimes tumors that are actually colored here based on the different cancer type but from different individuals cluster very separately. And actually, that's because of strong copy number variation that you observe in cancer that really drives a lot of the chromatin accessibility signal, because if you double the content of DNA on one chromosome, you also double all of the strengths of each of the peaks of chromatin accessibility. If we regress all that out and sort of reproject, we see suddenly now that the cancer type is actually driving the majority of sort of the variant, the clustering ability in this UMAP. And so you have bladder cancers clustering with themselves, and colorectal cancers clustering with themselves, suggesting that there's common regulation within cancer subtypes. So we can do the same trick where we can predict open chromatin accessibility from the DNA sequence itself and interpret then which bases are important. And here, for example, are 3 different types of breast cancer: luminal, HER2, and basal. And now we're interpreting these BPNet models trained on these data at this locus around top 2A. And you can see, for example, that in all of the luminal samples here, there's this strong FOXA1 motif that's popping out, but not so much down here in these basal cancers. Whereas this SP1 motif is universal across both the luminal and basal cancers. And that's actually consistent with the gene expression we observe. FOXA1 is high really only in these luminal cancers, and SP1 is high in all of these cancers. And of course, the model knew nothing about the gene expression, but it's accurately predicting sort of the activity of the FOXA1 motif in the cell types, in the cancer types that have FOXA1 high expression. But now we can also try and interpret somatic mutations, not just germline mutations that we've shown previously, but these are somatic mutations that are occurring in cancers themselves, drawn from these large whole genome sequencing cohorts of cancer, whole genome cancer sequencing. We can plug them into our model and ask which cancer mutations cause large effects on chromatin accessibility, and thereby maybe driving cancer phenotypes. And we actually have used this interesting in silico mutagenesis approach, where we not only install the mutation we observe in cancer, but we actually ask how strong is the motif that this mutation is actually ablating, and use that as our score. And again, now what we're looking at here is the enrichment of these somatic mutations near cancer-associated genes versus away from cancer-associated genes. If these mutations are causal, you might expect them to be in regulatory elements around cancer-associated genes. And if we just look at all mutations, there's really no enrichment at all. If you look at mutations in peaks of chromatin accessibility, there's weak enrichment. But if instead we use our model to prioritize which mutations are highly impactful on the motifs that they're ablating, you can see that there's a pretty strong enrichment for mutations that have this large effect size on chromatin near cancer-associated genes as opposed to away from them, suggesting, I think for the first time, some of the first evidence that non-coding elements can be mutated in cancer and have a functional effect. And we can, of course, interpret these as well. For example, here, this is a FOS motif around the TET2 gene, and this is the base that gets ablated, and it's predicted to just destroy this FOX motif and strongly change the chromatin accessibility of this peak here. Okay, I just want to talk really briefly about some other work in progress where we're really trying to build these models of chromatin regulation across sort of intermediate fetal human development. So we've collected almost about a million cells from about 76 different samples across 12 organs, profiled in this case with simultaneous ATAC-seq and RNA-seq. Here's across different gestational ages and all of these different tissue types in human fetal tissue. And our goal is to really develop models that can assess the regulatory impact of any genetic variation on the non-coding regions across human development. And this is driven in collaboration with Illumina and the Kundaji lab, as well as Sel and Sam and Betty in my lab. And I'm going to go through this pretty quickly. So I understand this is a complicated diagram, but you're really this is just a clustering of all the different cell types. We observe about 203 different cell types in all these different tissues. And of course, if we clustered them based on gene expression, they cluster out in the reasonable ways where there's neuronal cells and immune cells and cardiomyocytes, endothelial cells, et cetera. We've named all these and annotated them with expert annotations effectively. But again, we're going to try and train these neural network models on the chromatin accessibility of these different cell types. We're going to identify which bases are important. And then we're going to try and identify collections of motifs de novo to try and identify all the possible motifs that are occurring in human fetal development just from the chromatin accessibility and the inferred importance scores of the different bases that are trained from this model. And what we get are sort of 5 different classes of motifs. We get sort of a simple base motif, like for this ETS factor, and we get maybe a simple motif with a couple base pairs nearby or in the flanks. We get homocomposite motifs where you have 2 motifs of the same factor. We get heterocomposite motifs where we have 2 different motifs next to one another. And we get sort of a collection of other things as well. And in general, we see 92 different classes of these sort of simple motifs. Maybe if you combine them with base with flank, maybe that's 150. And then more homocomposite and heterocomposite motifs. Actually, the really interesting thing is most of these motifs are predicted to be increasing accessibility, and a relatively small fraction of them are predicted to be repressing chromatin accessibility, which I think we're really interested in as well. Just to give you a sense of what this looks like, here are on the left column here, we're showing all the sort of the weight matrices of the bases that are important have been identified in these de novo motifs, and then the most similar known position weight matrix from the existing compendiums of motifs, the number of instances we identify in all of our different cell types, and the number of different classes of similar motifs that we are sort of clustering together with this name. And, you know, like, for example, there's 2 different kinds of one-cut motif, and they're very, very similar. They actually vary only by whether or not you have a T or a C here in this middle base. That's sort of what these different classes are. And then we can organize these basically on based on their entropy. So how many different organs or compartments that they're identified in. And you can see that there are whole classes of transcription factors that really appear in every cell type that we look in. And then there are some that are much more cell type specific, appearing much more, for example, in liver or thymus, and sort of much more cell type or tissue type, for example, immune cell type specific. And so we can do the same kind of thing with these homocomposite motifs, and you can see again de novo the motifs over here, and then here the nearest known position weight matrix. And so this is trying to identify these motifs that occur in sort of a bunchy way. And actually, there's a lot of structure in here as well. So when GATA motifs appear together, they can appear in a head-to-head configuration or a head-to-tail. Sorry. This is head-to-head, this is head-to-tail, and with different spacings between them, and we can try and understand if there's actually more subtle subgrammars within these different orientations. And then we have these heterocomposite motifs. I think one of the things we're excited about here, for example, is we've already sort of recapitulated here. There's 500,000 different instances of this sort of heterocomposite motif, neuro-D and a BHLH factor. And one of my colleagues from Stanford, Ioana Wysocka, just recently published this. She calls it the coordinator motif, which plays a really important role in neural crust development. And so this is one of the many sort of heterocomposite motifs we've identified. This one is dominant actually in skin, and in this case, brain, sort of ectoderm. But we're actually also really interested in some of these other motifs we've identified here to see if they play a similar important role in this the coordination of regulatory elements in development. Okay, I'm going to skip that. But the one other thing that I'm really excited about is that there are Actually a relatively small collection of transcription factor, or I should say motifs that are predicted to decrease chromatin accessibility, and that's shown here by these bases pointing down, right? They're contributing negatively to the prediction of chromatin accessibility. And some of them are incredibly common. So 33 million examples of this ZEB1 motif that's predicted to decrease chromatin accessibility, and they tend to be really universal across all tissue types. And you can just sort of show this here, all these different cell types, the proportion of repressive versus activating motifs. So while there's lots and lots of different sequence variation in the activating motifs, they're only about 2/3 of these elements, whereas a full third of them are these repressive motifs, but those are only drawn from a really small number of different motifs that we observe. Like there's only 12 or something. Okay, and then finally, there's all of these different motifs that really don't clearly match known motifs, and we're really excited to try and dig in to see if, for example, these some of these unresolved factors that occur millions of times across different tissue across different cell types, whether or not we can link them specifically to unresolved to transcription factors that maybe are unknown to science, and maybe prove out some of these elements just from the de novo sequence motifs. And then, of course, we'd love to prove some of these features out in sort of tractable human model systems. So we're collaborating with Suju Pasca and Casey Gifford, who have heart and brain organoids that are really recapitulate a lot of early human development, so that we can try and sort of validate some of our hypotheses that we've generated from these data sets. And if you're interested in this, please give me a call. This is sort of ongoing work. Okay. So that's the chromatin accessibility story. Now I'm going to switch gears to try and get at exactly what's happening at these regulatory elements that are being bound by different transcription factors, and try and build up sort of quantitative biochemical models that might explain how different transcription factor binding sites, for example, if we have 2 transcription factors or 4 or 8 here, bring about gene expression. And for 30, 40 years, it's been known that there's a lot of nonlinearity between the number of transcription factor binding sites and the output of gene expression. So where's this nonlinearity coming from? It could be because there's cooperative binding of the transcription factors to these different binding sites. You need sort of 4 before you're able to bind enough and evict nucleosomes, for example, and then drive gene expression. Or it could be that you get, for every transcription factor binding site, you get the binding of 1 transcription factor, but you need 4 or 5 or 8 of these before you can activate the promoter, right? So that the cooperativity is occurring at the level of the promoter activation. And in order to really understand how this is occurring, what we would really love is the magic camera, right? You can observe a lot by watching. According to Yogi Berra. So if we could actually measure which transcription factor binding sites are occupied by a transcription factor, and then also relate that to the gene expression or the molecular features of the promoter itself, we might be able to really build a quantitative model that tells us how we go from the number of transcription factor binding sites to the actual occupancy of transcription factors to the actual gene expression state. And so this is work driven by 3 really talented graduate students in my lab, and Lakra Bintu, a close collaborator here at Stanford. So yeah, Michaela, Julia, and Ben, and Lakra. Okay. Okay, so we've built this tractable synthetic system to really study this transcription factor binding site synergy, where we've installed into a human genome different numbers of TET-O binding sites here, so from 1 to 9. And then we express this RTET-RVP48 synthetic transcription factor, which can be recruited to these transcription factor binding sites with the addition of doxycycline. So we can dump in dox, recruit this RTET-RVP48, which is going to drive the expression of the citrine gene through this MIN-CMV promoter. And as we add more and more binding sites, you can see we observe this super cooperative expression where you need to get to 4 or 5 transcription factor binding sites before you see really strong effects on gene expression. So why? Well, we're going to try and understand that by actually doing single molecule footprinting that will identify the occupancy of these microstates. So let's say this is a transcription factor binding site. This is a promoter. You might expect there to be a lot of different molecular states. There could be nucleosomes occluding the promoter or the transcription factor binding site. You could have a transcription factor bound here, but an open promoter, etc. So what we're going to do is use this method that was pioneered by Arno Krebs and others to come in with a GPC methyltransferase and spray paint methyl groups onto regions of the DNA that are accessible, that are open. And so we're going to spray paint these little white circles on the ones we can. The ones that are bound by nucleosomes, we can't, so they're going to stay black. And then the ones that are bound, for example, by a transcription factor, they're going to stay black too. And then we're going to read out the occupancy across these single molecules using methylation-based sequencing approaches. Okay, so we're going to get out full reads, but whether or not that GPC was methylated or not, we'll be able to determine through the sequencing and then build sort of molecular level footprints. And so what this really looks like on individual molecules here is this. So here we have a molecule with 5 different transcription factor binding sites. White is not blocked, black is blocked. Here's the promoter. And I claim that I can immediately actually unambiguously assign the molecular states to these different molecules. For example, here there's 5 transcription factors bound. Here there's 3 bound and 2 unbound and a nucleosome here. Here there's 1 bound and a nucleosome and then another nucleosome. And then here all nucleosomal. So I claim this is really as close to that magic camera, that camera that can resolve molecular configurations as we're going to get. We can sort of actually build up the microstates of the different regulatory elements using this approach. So what does this look like? Now I'm switching to a different view of this. We have 8 different transcription factor binding sites here. Now every row is a different molecule. And if I'm protected, I'm black. And if I'm unprotected, I'm white. So you can see at 8, these stripes, which are the binding of the transcription factors at each binding site. But we, of course, have this, we have this picture from 0 transcription factors all the way up to 8 transcription factors. And you can see as you increase the number of transcription factors, you kind of start to open up here at 4 and 5, where you see more and more clear binding. And then 6, it's quite clear. And if we quantify that, we can look at the average transcription factor occupancy as a function of the number of binding sites. And you see this cooperative effect, demonstrating that the cooperativity, at least to some degree, is on the binding side here. But we get not only the average occupancy, we also get all of the distribution of molecular substrates. So if we look, for example, with molecules of 7 transcription factor binding sites, we can see some of them have no transcription factors bound. Some have 1, 2, 3, 4, 5, 6, 7. And it's a nice wide distribution. So can we fit this with a really simple competition model, right? Where it's just mass action, it's just the concentration, or there's an energy of binding of the transcription factor, there's an energy binding of the nucleosome, they just compete with one another, call it a day. That would be very simple. And it turns out this doesn't work. This doesn't recapitulate either the cooperativity we observe in the number of binding sites or this bimodal behavior we observe in the occupancy of different transcription factors for a fixed number of binding sites. So that doesn't seem to work. But we actually know that VP48 recruits other cofactors. And these cofactors are things like chromatin remodelers that might, we imagine, have an effect on the off rate of the nucleosome or the energy binding of the nucleosome. So if we just assume that once you've recruited 1 transcription factor, you have the potential to recruit cofactors that then reduce the binding energy of the nucleosome. So this is one more free parameter, which is this just the reduction of energy associated with the nucleosome once I've bound 1 transcription factor. This actually fits the data basically perfectly, right? So we recapitulate this bimodal behavior, and we also recapitulate this sort of cooperative effect here in green. So this nucleosome destabilization model is what we have sort of arrived upon. Okay, so what about the promoter states? Well, we can look at this with 8 transcription factors bound. Here's the Tata box, here's the TSS, and we're going to split these molecules into molecules where there's no nucleosome covering the transcription start site versus nucleosomal molecules where there's a nucleosome that's subtending this TSS, and therefore we're going to call it inactive. And it turns out this fraction of active versus inactive is also correlated with the gene expression we see at the end. Okay, but as you can see, as you increase the number of transcription factor binding sites, this fraction of active molecules also increases sort of in a cooperative way as well. But so now we can actually relate not just the number of transcription factor binding sites, but rather the transcription factor occupancy to the number of active promoters. And it turns out this is a pretty simple relationship. As you increase the number of actual transcription factors bound, you get basically a linear increase, or it can be described as this linear activation model, where the probability of turning on the promoter is linearly related to the number of transcription factors bound at the enhancer. So that means there's some sort of potency of every transcription factor, and as you add one more and more, they linearly contribute to the likelihood that you turn on your promoter. So there's no real cooperativity there. It's all sort of linear in probability. So now we've separated these two effects into an occupancy component and a potency component. So the question is, basically, how many transcription factors do you have bound at your enhancer element, and how potent are they? And if I multiply these two together, I might expect that to bring about my gene expression. And we can also independently modulate these two effects, right? So if I change the effective transcription factor concentration by changing the tetracycline concentration, which changes what fraction of the synthetic transcription factor is able to bind, I can change sort of this occupancy curve. But once I'm bound, okay, so again, I'm changing the occupancy curve. I'm changing the average number of transcription factors occupied as a number of binding sites. As I decrease the concentration, that comes down. That makes sense. But if I plot the average TF occupancy, that is to say, how many transcription factors are actually bound at the enhancer versus the fraction of the promoters that are active and expressing, that is not changing here, right? Because the potency doesn't change. It's just harder to get on. But once I'm on, I'm just as good at activating the promoter. Okay? So that means tuning this concentration changes this occupancy, but not this potency. Whereas if we inhibit these cofactors, we can actually affect both, one or the other. If we inhibit BAF, so this is that chromatin remodeler, suddenly we get less binding. So the transcription factors have a harder time getting onto the chromatin because they have a harder time evicting those nucleosomes. But we also affect this potency. So now, once I'm bound, I have a harder time activating the promoter too, because I'm presumably unable to evict the nucleosome at the promoter and drive it. P300 is an acetyltransferase that's also implicated in gene regulation. But if we inhibit P300, we really don't have an effect on binding, it turns out. But we do have an effect on this potency. So P300 is important not for getting on and occupying the enhancer state, occupying my binding site as a transcription factor, but for driving gene expression. Okay, so again, our model is that we can, if we live here, we can modulate either the occupancy of a transcription factor or its potency by changing the concentration of the transcription factor. We move along this occupancy. By changing P300, we can move along this potency, and BAF moves us both directions. And the overall expression is just this potency times this occupancy. And that actually models this cooperative behavior quite well overall. And actually, we also have a thermodynamic model that predicts sort of all of the molecular substates underlying all of these data. So that's a synthetic transcription factor. Does this actually work for sort of native transcription factors? And to ask that question, we went after this really nice endogenous response system, the interferon response system. So in homeostasis, it's thought that STAT2 and IRF9 sort of bind these interferon response elements, but don't drive gene expression of the interferon response genes. Whereas if you add interferon, there's this phosphorylation that occurs that phosphorylates STAT2 and STAT1, which then dimerize, bind IRF9, bind the interferon response elements, and drive gene expression. So in order to play around with the system, we did the same thing. We installed different numbers of these interferon response elements upstream of the citrine promoter, sorry, this citrine reporter gene driven by this MinCMV promoter, installed that into the chromatin at a safe harbor locus. And as we add interferon, we see really strong induction of expression of centrifuge. But now we can look at the footprints of these interferon response elements without interferon. And so without interferon, you actually see chromatin accessibility, and you see these narrow footprints where actually we posit that STAT2 IRF9 is actually bound, but the nucleosomes, but the promoters themselves are basically all nucleosomal. They're all overlapped by a nucleosome, and there's no gene expression. Whereas if we add interferon, suddenly we get fat footprints across all the transcription factor binding sites, which is consistent with the STAT1, STAT2, IRF9 trimer binding the interferon response elements. And suddenly we see a bunch of activity of the promoter. A bunch of those nucleosomes are evicted, and we also see gene expression. And it turns out we only really see these wide footprints as a function of the interferon response element motif copy number upon induction of the interferon, whereas these narrow footprints actually don't really change. And if you look at the gene expression, it correlates really only with the number of wide footprints and not with the number of these narrow footprints. And actually, we see more or less all of the same kinds of behaviors we saw with our synthetic system. If we look at the gene expression as a function of the interferon response element number upon interferon stimulation, we see this cooperative behavior as we had previously seen. We can also try and calculate this this potency. And here, the dotted line is our RTED-R VP48. That is to say, the fraction of promoters active as a function of the average number of footprints. And for our fat footprints, we see actually really strong potency for these guys. And as I've said, the fraction of active promoters correlates really well with this gene expression by flow as well. So what can we say about the dynamics of these different, can we say something about the dynamics of the induction and the chromatin regulation here, both in our synthetic system and in our and in the interferon system? Well, we can just do this experiment at different time points after induction, in this case for doxycycline in our synthetic system. And as we do that, we actually see that at 0 hours, almost no transcription factors are bound, and it takes maybe 24 hours to reach this steady state where we have this distribution of different transcription factors bound, and it doesn't change over time from 24 to 48. And if we look at the number of transcription factors bound, this sort of stabilizes pretty quickly, but the fraction of promoters active actually takes even longer to stabilize. So there's a delay between getting onto the chromatin and driving the promoter to be active. And that's consistent because we see this sort of observed potency increase in its slope as a function of time, again, sort of plateauing here at maybe 24 to 48 hours. So based on all this, we can build a really relatively simple model where we have our thermodynamic model for describing how many transcription factors are going to be bound. And then we just have simple sort of one-state kinetics that generate transcripts, and they go to decay, protein decay and mRNA decay independently. And with this relatively simple kinetic model, we can explain sort of all of the intermediate molecular states. We can explain this transcription factor occupancy over time. We can explain the fraction of promoters active over time. We can explain also the amount of RNA expressed, which of course takes a little while after the promoters open. And then we can also explain the protein, which of course takes a little while after the RNA gets expressed. And we can do this actually not just for, we can do this for all different transcription factor binding site and over all time. I'm showing the data on the top and the fit model on the bottom. So I think we really understand all of these kinetic parameters. And if we look at these interferon response elements, they're actually much faster than these synthetic RTED-R, mostly because they're able to bind faster, we think, and also activate faster. So as you can see, like the wide footprints come on, within 30 minutes or so, and really, really saturated after 3 hours. And actually, the most important thing is over here, this fold change relative to our TET-RVP48. The time T1.5 to saturate my binding is much faster for this ISGs. And also, the kinetics of the promoter activation are also much faster for the ISG. So we think that because the ISGs are meant to respond to sort of infection, they really have to activate very quickly. So both the chromatin accessibility state is sort of pre-formed because the dimers are bound there and the chromatins are already accessible. And all you have to do is phosphorylate and then bind with the trimer to drive expression. So you don't have to evict all of these nucleosomes and have this slow timescale. But not only that, but it looks like the promoters are sort of ready to be fired as well. Either that or the ISG, the trimer itself is just very, very potent and is able to drive nucleosomes off the promoter very quickly. So I think we're sort of uncovering sort of the underlying logic of how to get response elements to go fast. Okay, so the last little piece I want to talk about, which is sort of unpublished, is how can the sequence context affect transcription factor binding and unbinding, and can we get even more quantitative with respect to our understanding of the transcription factor affinities and use those affinities to predict chromatin state? And for example, can we quantitatively measure the affinities of these transcription factors in vitro and then use those to predict the in vivo behaviors of regulatory elements? That's sort of the reductionistic, like promise of sort of molecular biology and biochemistry, right? Can we measure fundamental constants of these different proteins and use them to predict the behaviors of cells? Okay, so what we're going to do is characterize the binding kinetics of these mammalian transcription factors in vitro for lots and lots of different motifs, so we can really understand the sensitivity of the binding to these different motifs, and then build models and compare these models to in vivo footprinting data. And this is again driven by Julia Sheppy and Emil Markland, who's now has his own lab in Sweden. So we're going to use this high throughput measurement device that we've developed in our lab that repurposes a MiSeq flow cell and uses that on a high throughput imaging station. So after you've sequenced an Illumina flow cell, what you have is a post hoc DNA array that has hundreds of millions of different spots of DNA that all have that are about a micron in diameter and maybe 1,000 different identical molecules in each spot, which you've sequenced. And so now you can, for example, flow in fluorescently labeled protein and ask about the binding kinetics of that protein to, 10 million different sequences. And so the way you do this is you build in variable motifs, you sequence your DNA, you strip it, you regenerate the double-stranded sequences, and then you flow in this fluorescently labeled protein. In this case, we're looking at KLF1, a DNA binding domain. KLF1 is a sort of a classic eukaryotic transcription factor. And as we add, if we add 100 nanomolar KLF1, we can watch different spots light up and watch the kinetics, the on rates of these proteins onto the different sequences. And then we can also flow out KLF1 and watch the dissociation. So what does that look like? Well, here is the binding to sort of a canonical motif here. Here's what the sort of fiducial mark looks like. It doesn't have any, and then the negative control doesn't have any binding. And we can also watch the off rates here. And we can use this then to build up an energy landscape for binding across the motif. So what this is the relative binding energy if I mutate any base along the canonical motif here along the diagonal, and then any double mutant here in the full matrix. And of course, we have triple mutants and more than that as well. So just as some background, it's been thought actually in bacterial transcription factors, the transcription factors transcription factor binding obeys this sort of three-state model where transcription factors can diffuse get into this non-specifically bound testing state, and then specifically bind to the target motif, or end up dissociating. Now, this is distinct from sort of a two-state model, which would predict that association rates are sort of constant, and dissociation rates are the thing that changes in order to get changes in affinity. This is the two-state model. This three-state model actually predicts a tight coupling between the association rate and the dissociation rate. That is to say, changes in affinity come from both changes in association, macroscopic association, and changes in macroscopic dissociation. And that's really what was observed for bacterial transcription factors, this coupling between the association rate and the dissociation rate. So it's much more sort of, there's correlation across these two. It's not as if all of the change comes only from dissociation and the macroscopic association doesn't change. So we see both changing here. And actually for eukaryotic transcription factor, in this case KLF1, we see the exact same thing. Now, again, we're plotting the macroscopic association rates versus the dissociation rates. So we can see strong coupling between the two. We're looking here at both motif mutations in red and also mutations of the flank sequence. So keep the core motif the same and only change bases outside of the flank. And it turns out, outside of the main motif. And it turns out that has huge effects on the association and dissociation rates and the affinity, therefore, too, right? And so what we can do is if we block reassociation, we would expect all of these macroscopic rates to collapse to the microscopic dissociation rate, which really should just be a line here, which is actually what we see. So if we add huge amounts of dark competitor, basically we collapse to the microscopic dissociation rates here, which is sort of supporting this idea that there is this intermediate state that isn't necessarily occluding the transcription factor binding site. And if we add a bunch of dark competitor, this intermediate state can no longer rebind. Now we get the overall off rates and we get much faster overall dissociation when we eliminate that rebinding pathway. Okay, the other thing we can see is as we keep different amounts of sequence constant around the motif, we get different amounts of variation in the association and dissociation rates. So if we have just totally variable flanks, we get huge amounts of variation in the on and off rates, even if we keep our motif constant as the core canonical motif. Whereas if I fix more and more bases, I get less and less variation. So it's really being caused by these variable flanks. And we can build a simple model to predict the delta G's associated with all of the different bases in the flanks. And this model predicts unseen data really, really well. So we think we have a really nice, relatively simple model that can predict the affinity of this KLF1 based on just flanking changes. And then we also wanted to determine whether or not we can measure the KLF occupancy in cells, because we have all this kinetic information in vitro. Can we get kinetic information in vivo using our footprinting approach? So what we did was we built these different tandem arrays of different transcription factor binding sites. And again, this is KLF1 binding sites, either the consensus motif or a broken motif. We measured the kinetics of binding to these different things in vitro. Again, here's what the kinetics looks like. And then we installed these guys into cells. And now, again, we put these into safe harbor locus. We're going to look at KLF1. KLF1 in these K562 cells is the dominant transcription factor bound, and all these other KLFs are also expressed, but they have basically the same DNA binding domain. So we're kind of looking at KLF DNA binding domain dynamics in these cells. And so what we can do is methylate for different amounts of time now. So if we have two transcription factors bound and we methylate, we would expect these guys to protect the methylation. But if we methylate for longer and longer, they might fall off, dissociate, and then we will end up methylating these. And at infinite time, everybody's fallen off and we'll methylate every base. Does that make sense? And this is actually exactly what we see. If we methylate for 2 minutes, we see these nice stripes of protection where the transcription factors are bound. But if we keep methylating for longer and longer, eventually we just methylate all the bases, which means we're getting information about the off rates of these transcription factors in cells using this approach. And so we can build a nice model to try and estimate that and actually calibrate our methyltransferase measurements using CTCF as a model. If we methylate for different amounts of time, you can see we methylate all the way to saturation here in about, say, 4 minutes, maybe, of methylation time. And so we can use this rate as well as our predicted off rate to, well, first of all, this is the methylation rate. It fits really nicely to sort of single step kinetics. So we have maybe a time constant of 2 minutes or so. And then we can measure all of these dissociation rates at the transcription factor binding sites, and then fit this to a model that takes into account both the dissociation and the time scale for methylation. And we get an off rate that's about 1.5 minutes, which is actually basically exactly what we observe in vitro. So we've observed off rates that are basically identical in vitro and in vivo, which is very satisfying because there's been a lot of sort of questions about these different rates. Okay, I'm going to go a little bit faster because I'm almost out of time. We can also, now that we have this full thermodynamic model for the binding affinity of a nucleosome and the transcription factors, we can actually predict what's going to happen if we have variable spacings of transcription factor binding sites. And here's our model predictions for the number of motifs and the average occupancy, as well as the nucleosome occupancy across these different motifs. And you can see that we really do, I think, have a handle, at least almost quantitatively, for exactly what we would expect with respect to nucleosome occupancy as a function of where the motifs are in space. For example, if you have tighter motifs, they are better at evicting nucleosomes, and if you have them spread out, they have to compete against more nucleosomes, so they have a harder time getting onto the DNA. Okay, so the last thing I want to mention is, remember, we had all this information about the flanks in vitro. If we install weak flanks all the way to strong flanks in vivo, we see that the weak flanked motifs, even though the motifs are perfect, the flanks make them so that they don't bind at all. Whereas we get really strong flanks, we see really, really strong binding, as you might expect. But we can kind of model the fraction of transcription factor bound as a function of this in vitro delta delta G and get reasonable concurrence across these things. And also, if we change the concentration of KLF1, we can get this binding curve to shift to the right, showing that we can use mass action to push more KLF1 onto these weaker binding sites. So we're sort of building up from pieces now a sort of quantitative biochemical model of how chromatin functions in cells based on these fundamental biochemical parameters. Okay, so I'm going to skip this and just say that collaborators are really central to all of my science. It's been wonderful to work with Howard and Anshul on a lot of the stuff that we talked about earlier, and then Lakra on a lot of the sort of quantitative modeling of chromatin architecture and synthetic systems. And I want to acknowledge my lab as well as funding sources. And sorry for going probably a little bit over, but maybe there's a little time for some questions as well. Thank you.
Thank you so much. That was awesome. I think you can see we do have a few questions in the chat.
Okay.
I don't know what the easiest way to do it. Do you want to maybe just read the question?
I don't see the chat. I guess I'll stop sharing and find the chat. Let me.
Okay, it's right at the bottom. It should be right at the bottom.
Yeah, I see it now. Yeah. How do you imagine that BAF can change both potency and occupancy? They seem like different quantities. I don't think so. I think that once you can imagine that binding, if I have BAF, I can help evict nucleosomes near me. And that opens up other sites for me to bind other more transcription factors. So that's a natural way for BAF to help you bind. And you can also imagine that BAF, if I'm at the enhancer and I happen to bump into the promoter, which is actually really close, I can also evict the nucleosome that's over the promoter. And if I evict that nucleosome, if that is the rate-limiting step for this promoter, or at least a rate-limiting step, then I also help transcription go, because now the Pol2 can bind and everything can start to fire. So I think that they're more related than one might initially think. There have been a recent publication by Brahma and Henikoff in Nature Genetics where they propose a model of BAF, Pol2, and TFs combining to remodel it and evict nucleosomes. In this model, BAF would help TFs bind. Are you aware of the model? Do you think it is in line with what you found proposed? I think that how recent is this? Maybe maybe I need to look at this really carefully. But I think that people have also thought about this. Like, I don't think it's crazy to imagine that BAF can help. There's a lot of data where people knock down BAF, or and there's lots of effects that sort of happen very quickly, which is consistent with these it's consistent with BAF helping things both at distal elements and at promoters. So I think that the model is consistent with lots of other data as well. I guess I didn't know Hennekoff had a specific model where he talked about this. I guess I'll take a look at that paper. That's great. We've been focused on getting the rebuttal in, so maybe how are noises over time depending on the number of bound TF? I'm not sure I understand that question. But if you want to rephrase it, I don't think I'm happy to try and answer it. In addition to cooperative TF interactions, have you explored competitive or antagonistic interactions? Yeah. So we started with a really simple synthetic system and homotypic transcription factor binding. We will use this system, I think, to complexify things. Once we really understand the simple case, I think then we can add more and more different binding sites or sites that are recruiting different transactivating factors that may have competing influences and things like that, and really try and build up a quantitative understanding of how these things influence one another. Any other questions? You can just unmute and ask. Yeah.
Yeah, feel free to turn on your mic and throw questions out there too.
How long in base pairs is like 4 motifs? Is it like 100 base pairs?
Yeah. I think these motifs center to center distance was 22 base pairs.
For each one?
Yeah. So 22, 22, 22. Yeah.
So it's like
either 19 or 22. We did some different spacings depending on what we had to do.
So it was almost like half a nucleosome is like the critical threshold to evict a nucleosome. It's like what you're
something like that. I mean, I guess I didn't show this, which I probably should have. We also chopped off the activation domain and just looked at the DNA binding domain. And it's way worse at evicting nucleosomes, as you might imagine, because it can't recruit BAF and do all the eviction stuff. And it is well fit by a competition model, like a straight up nucleosome binding energy versus transcription factor binding energy model. Because there's no magic that it's bringing in anymore, right?
You almost like have like echoes of like a Hill equation or like cooperative binding on oxygen to like hemoglobin. That's what it looked like when you were.
That's exactly that's right. That's but the cooperativity is through the effects on nucleosomes, right? It's not. I didn't show this either, but if you look, I should have showed this. If you look at the binding of if you look at all molecules with 2 transcription factors bound, but 5 transcription factor binding motifs. If binding is independent, you expect them to just be totally random, independent binding. And that's exactly what we see in fragments of DNA that don't have nucleosomes. But if you add in the nucleosomes, then they tend to be clustered because there's all this binding energy of the nucleosomes that it gets, right? It prefers for the nucleosomes to be sort of on the DNA, and therefore it looks like there's cooperativity. But if you just look at pieces of DNA where there are no nucleosomes, the binding's totally independent. So there's no cooperativity through like protein-protein interactions or even like shapy interactions through the DNA. It's all through this eviction of nucleosomes lets me bind another factor over here.
Yeah, like the nucleosome is the intended default, and like you're forcing it away from that intended default, and you have like some threshold you have to reach to like push that guy off.
That's right, and you have to have enough so that you can get your beachhead where you recruit your nucleosome remodeler in, and then suddenly that's sort of the phase transition, right? So,
I have a somewhat unfair question because like my understanding of your system is you essentially have the binding sites and then you have the promoter and like
it's very proximal. That's right.
Well, I'm actually not worried so much about the distal. That's too hard. I'm not going to ask that unfair of a question.
Eventually, we'll get there. Yeah.
but what happens like, if you have that start site and then there's some spacing for the next element to be transcribed? Is that coherent? Like, is it, I have a fragment where the transcription factors are bound, then I have a start site and there's like a little like imaginary codon there. And then if I skipped a nucleosome and put another codon, that would be a really efficient way to get two codons linked together in space. Does that make sense?
Yeah. I mean, I guess the way I conceptualize, maybe this is wrong. The way I conceptualize the promoter is that there's like a TATA box, and we can see TATA binding protein. I didn't show this, but we can see the TATA binding protein by protection. We can see the transcription factor loaded, and then we can also see, and then I feel like there's like a pause state, and then it fires, and then I don't think about it anymore. Like it fired, if that makes sense.
No, totally. I understand.
And the nucleosome occupancy at the promoter, like that partitioning between like nucleosomal and non-nucleosomal promoters correlates with gene expression really well, luckily.
Yeah.
So that it's consistent with sort of that being the rate limiting step, although we really need to do really careful biochemistry about what really the rate limiting steps are for this min-CMV promoter. Obviously, it can be different for different promoters, but that's another area of investigation we're really excited about.
Yeah, I just meant because like if you think about like the genes in neurons, like those are huge genes,
Yeah.
right? Like
Yeah.
they don't just have like a TATA box and like the whole gene then gets turned on. Like you have to think like, oh, how do I deal with 100,000 base pairs of
Yeah, I mean, I don't know. Most people who think about gene regulation think once the polymerase is fired, its fate is sealed. It's going to make a transcript. But there's lots of evidence that's not true from Ali Shalafor. I mean, like, I guess he's maybe on here. I don't like that. Like, he really cares about elongation as a regulatory step, which I think very convincingly it is as well. It's just that, people focus on the things they hope to think about. And, in this case, you're just expressing a short, like, reporter gene, right? It's really relatively simple. And I think it must have one splice site or something. It's really simple, right? So,
yeah. Thank you so much. That was great.
yeah.
Okay. Any other questions before we start our weekend?
Can I ask a question?
Sure. Hello.
Hi. So my question. So large language models have been really important and huge development in like the machine learning field in the last couple of years. You guys have been really successful using convolutional neural networks. I was curious, like, what you see as like any potential of like applying large language models to your work, or the application of foundation models, and like what you could do with those that like maybe you can't do currently.
Yeah. I'm going to say some controversial things here. I mean, large language models make a lot of sense when the inputs and outputs are identical, if that makes sense. So, right, like language goes in, language comes out, and it's a generative model, and that's great. So I guess I just don't understand, like DNA sequence would come in, and then DNA sequence comes out, and I guess you can make synthetic open chromatin regions or something that way. Is that I mean, you would learn just on the DNA. People have definitely done this and tried. And I don't know if it's been published yet, but I think all of these models do worse than the models that are trained on the underlying structures. Like if you want to learn about protein sequence, you should train on protein sequence. If you want to learn about like it's a little hard to understand how LLM specifically will be super useful here. And then as far as the universal embedding, like these other models, I have not seen clear demonstration that they're better than PCA, frankly.
Totally hear you.
They're very strange to me. I really don't understand their utility at all yet. I haven't seen any evidence that like they can predict perturbations. That would be a minimal thing you would imagine that maybe they could do, but I don't understand them yet in terms of utility. I think that there is a world where they can start to be useful. But again, I think all the molecular sub-information, like the cell is a physical object. So we're not trying to like model language. We're trying to like model like the trajectory of a ball going through the air. Like it's a physical reality we're trying to figure out. So I think giving these models more information about what we already know must be causal and the intermediate molecular information will for sure help them. So I don't think we can take this approach of just like, oh, we can just let the models learn everything. Everything's going to be fine. I don't think that's going to work.
Thank you.
That's like my philosophical. Yeah, okay.
All right.
Okay, sorry, I went way over.
No, it's all good. Sorry we made you use your voice so much.
No, I'm still talking at the end. That's a good sign.
That's right. Okay, well, if there's no questions, I think we can let you go. Thank you so much for doing this. That was great.
Sure. Happy to
Appreciate it.
answer any other questions if you, I can stay on for a little bit if anybody else has any other.
Okay.
Or not. Whatever, that's fine. You can just like actually, unmute yourself and talk or whatever.
I do actually have another question.
Okay, cool.
So you built these convolutional neural networks, and then you functionally like interrogated those networks to sort of identify key transcription factor motifs, right? How do you establish like confidence in the findings of the model? Like, how do you, when you do like a regression, you have like confidence intervals, and you can say that something is statistically significant. Like, can you explain to me, like, is there an equivalent to your approach, or like, how do you determine like what, is a statistically significant observation and like what isn't using your approach?
Yeah, it's a really good question. I think that, I didn't show any of the figures of merit with respect to the model, like, Shannon, there's a bunch of ways of showing that the model does a good job at predicting unseen chromatin accessibility data. So that gives you confidence that the model is learning something real. The other thing people do is they train 5 models, and then you can ask about 5 models on different parts of the genome, right? And then you can ask about consistency there with respect to the magnitude of the effect. But a lot of the things we're doing are mostly rank order stuff, right? Where, the thing is predicting really big effects and really small effects, and we sort of trust big effects. I think I think it's a really hard question to answer. I think that you probably have to start wiggling bases and then saying like, oh, when you see a fold change of 20%, you also see something 20% in real data. But if you see a fold change of 5%, then it's hard to see in the really real data. I mean, you can kind of get that out of the QC metrics, right? Where, you see like 0.8 correlation with the chromatin accessibility peaks and stuff. So you might, there's ways to like say like, oh, we think that this is predicting things to within 20% or something like that. But I agree with you. I think that it's underdeveloped exactly how to think about reproducibility and stuff like that. A lot of people just train 5 models, say the models all sort of say the same thing. We're going to average them all together. They're learning something real. And then it really depends on how you use these models in the future, whether or not you really need to have the statistics, or you just use it as a pre-filter and say like, look, the top stuff's probably good. We're going to use that, right? It's a good question though. Yeah.
Okay. Hi, can I also ask a question really quick? Technical question. It was great, by the way. I was wondering if you have to worry about transcription factors dissociating in your cooperativity study. So do you have to correct for this, or if so, how do you correct? Have you cross-linked, and could you compare your data plus minus cross-link and see the same results, or something like this? So how do you go about stuff potentially dissociating?
Yeah, it's a great question. So this is all unfixed. So for ARCSET VP48, you know how I showed if you methylate for longer and longer, you eventually see the off rates, right? Because everything becomes accessible and you don't see the footprints of the transcription factors anymore. If you do that same experiment for ARCSET VP48, there's really no loss. So the off rates are slow for that synthetic transcription factor. For the other, we did something similar for the interferon response elements, and they're slow. And we also worked on our assay so we can methylate for like 4 minutes, like really, or 3 minutes, or really, really short. And we dump in huge quantities of the methyltransferase, and then it happens really fast. I mean, because you want it to happen instantaneously, right? And like that's the snapshot. So we don't see a lot of sensitivity for between sort of like the 2.5 minute and the 5 minute methylation. And so we're pretty confident we're capturing the occupancies pretty well there. I think no matter what, even if the off rates are super fast, you'll be able to capture the nucleosome occupancies because the nucleosomes are very, very, very slow, right? And for a lot of these things, it's probably likely that if the sites aren't bound, if the sites are accessible, they're probably bound kind of stuff, right? So I think you can get a long way with like time series stuff. And also if things are just really, really fast, you can do a lot with like nucleosomes and then inferring the binding energies and stuff from the nucleosome. You can do a lot with that. But I agree, like what you want is things are really, really, really fast and really efficient.
Sure, yeah. But then if it's really fast, so how do you quench the methylation reaction actually?
Yeah, I think we just go straight into, like, once you're done methylating, you can just dump it into like trizol. Like, it's a good question.
Oh, I see. Okay.
We tried, so, but you like, as soon as you're done methylating, you can be really harsh, right? Like, because it's dead now. So you go straight into like SDS or something, right? So I have to. Good question. But we thought about that a little bit.
Last question from my side. So have you ever tried 6MA as a methylation mark?
Yeah. So 6MA would be wonderful, right? Because it gets you every A. The problem is it's inherently a single molecule readout. And the beautiful thing about the GPC methyltransferase is that it's super efficient, really fast. Like, and like it goes, I don't know if you saw, but that CTCF plot went from, like, the modulation depth is basically 90%, right? Like it goes from no methylation to fully methylated, right? So that's super powerful because then you can more or less trust every methylation event. The 5, what was it? The 6MA. The 6MA. It's inherently a single molecule readout, so the quality is way worse. It's the short answer. I think we are interested in other deaminases where you might end up with an actual base change. Like, base changes are killer, right? Because then you can use like standard sequencing and it's perfect, more or less, right? Whereas like these other modulations, like you have to do a single molecule readout, and then you gotta sort of read it many, many times, and then you're sort of limited. But I agree that you could probably do a lot of this stuff with the FibroSeq style like analysis, probably. And we're playing around with that too. But it's nice to be able to, all of this was sequenced on Illumina, right? So you can go crazy deep and it's cheap and stuff. So. But it is true, you have to engineer in your GPC sites so that they are next to your, I mean, we get to do that, right? Because it's a synthetic system. But it would be much nicer to just look genome-wide at all A's or C's or whatever. But then you also have to get something super fast, right? So, and that works on double-stranded stuff. That's also really hard. Anyway, I can go on and on.
All right. Thank you.
Yeah.
Ciao.
Okay, awesome.
Okay.
Well,
thanks.
If that's it, I think we can release you from your.
All right. Great. Thanks so much for the opportunity.
No, thank you for all the time. We really appreciate it. This was great.
Okay, cool. Bye, guys.
All right.